Hello, friends. Today's topic complement system in immunology. Let's start with it. So, first, we will discuss the general properties of complement. So, first, we have to understand that complements are part of innate immunity and they represent normal serum proteins. They are totally 5% of the total normal serum protein. And their level does not increases either following any infection or vaccination. It remains the same, and because they are protein in nature, they are heat labile. uh, they are non-species specific. They are present in sera of all mammals, birds, and animals, and complements from one species can react with antibodies from other species because they are non-species specific. The third and the most important thing, that complement can only fix or bind to bound antibody. So complement cannot bind to free antibody, complement cannot bind to free antigen. It can only bind to an antibody which is attached to antigen, and which antibody has maximum ability to fix the complement. It is IgM, followed by Ig3, Ig1, and Ig2. Ig4 does not fix the complement. The binding site or fixation site on IgG is CH2 domain, and on IgM it is CH4 domain. The complement has cytolytic action. It kills the cell, it lyses the cells to which it attaches to, and the main way by which it does is having membrane attack complex. It attacks the cell membrane of that cell and put pores or holes in it, so all the contents of cell will come out and cell will die. So then we have to understand that its system comprises of approximately 30 serum proteins. They're grouped into complement components. The complement components are named by numerals. There are 9 components, C1 to C9. C1 has actually 3 components, C1Q, C1R, and C1S. The properdin system and the regulatory proteins, they are named by later symbols. So overall, total 30 proteins are there. There are 3 pathways by which it acts or it gets activated. The classical pathway, which is also known as antibody dependent pathway because it is activated by antigen antibody complexes. The alternative pathway, it is antibody independent pathway. This is the exception where complements get activated without antibodies directly. And lectin pathway, it resembles classical pathway, but it is also antibody independent. So classical pathway is antibody dependent, alternative and lectin are antibody independent pathway. uh, I hope that some of you are struggling to remember these pathways, but this rule, which is in point number 7, is very important. And if you can remember this, then you have mastered all these pathways. I will come to this rule again once we will discuss all the pathways. That is, C4 comes after C1 and before C2 in the cascade. And the goal is to form the common pathway from C3. Now, the larger fragments which are formed in these pathways, because these pathways involve enzymes and enzymes will act on substrates, and the products will be, some of the products will be larger in size and some will be smaller in size. So larger fragment will join the cascade, it will continue the cascade pathway, and the smaller fragments, they exert the actions. There are regulatory system, there are, you know, inhibitors which will stop the functions of this complement proteins. There are inactivators which will permanently destroy the complement proteins, and that's how the body keeps a check on this complement system. There are pathogens who can evade this complement system. They have evasion mechanisms, so we will learn about that. And if there is deficiency of either complement proteins or the regulator proteins, in both scenarios we will have diseases. The If we talk about site of synthesis, the liver is the main site of synthesis where C3, C6, and 9 are produced. The other sites are basically monocyte and macrophages where C2, C4 is produced. In small intestine or GIT epithelial cells, you have C1, and in spleen, usually C5 and C8 proteins are produced. So after this, we will learn the complement pathways one by one in upcoming sessions. So first we have to understand and master these general properties to understand the complement better. In next video, I will talk about classical pathway, then alternative, then lactin pathway, and then other relevant points. So stay tuned.